Hi everyone, my name is Sonali. Today I'm going to be talking about ion exchange chromatography. So the theory of ion exchange chromatography is that it is a process that involves the separation of ions and molecules based on their charge. It's commonly used to separate biological molecules such as proteins, peptides, amino acids, nucleotides. There are definitely more, but these are just some that I listed. Separation is able to occur just because the charge of the biological molecule can be manipulated by changing the pH of the buffer used, and we'll see this in a future slide. This is a method that is often used in addition to other chromatography methods. This is often used as a form of purification and a means to separate the mixture even further. Moving on, ion exchange chromatography is a form of column chromatography, and we know that in column chromatography there's a stationary phase and there is a mobile phase. So the stationary phase is the solid part. It is also known as the matrix, and usually in ion exchange chromatography, the matrix is resin. So the resin will be covalently bound to charged molecules, and the resin can be agarose or cellulose, anything like that, and different types of matrices or resins will have different properties. For example, cellulose has a hydrophilic surface. So on the other hand, the mobile phase is the liquid portion of the column. It's also called the eluent. This is the liquid holding the sample, whether it be the protein, amino acid, nucleotide, peptide, whatever it is. It's also usually a buffer, and it ensures that proteins can stay in their native states. So this is a diagram of what the column should look like during ion exchange chromatography. As you can see, the mobile phase is this liquid back here. This is going to be the buffer, like I said before. We also have the stationary phase as these round solid circles. These circles are going to be covalently bond to the sample molecule, which you can kind of see back here. You can see that they're sticking to them. The liquid is just going to flow down the column, and it's going to contain the charged molecule in there. So moving on to the components of ion exchange chromatography, to start off, we have the pump, which is right over here. This is going to provide a constant flow of eluent through the injector column and the detector. So next, we have the injector, which is right over here. This introduces the sample into the eluent stream and also onto the column, which is right here. The column separates the sample mixture into individual components. As you can kind of see right here, both of the samples are separated. We then move on to the detector, which measures the analyte peak as eluent from the column. Usually, an electrical conductivity detector will be used in ion exchange chromatography. And then finally, we have the data system, which will just collect and organize data at the end. So, how does it work? So in the case of purification of a protein, an impure protein sample will be loaded into the column right over here. As you can kind of see, these red positively charged protein molecules will be bound to the negatively charged group of resin molecules, and these resin molecules will kind of be attached to the sides of the columns. A salt gradient will also be used as an elute to separate the protein from the resin. So over here, in a low salt gradient, so not a lot of salt, few positively charged molecules will be eluted from the column, as you can kind of see right here. But when there is a high salt gradient, most of the positively charged protein molecules, which are down here, are going to be eluted. And this is because the salt separates the protein from the resin, like I said before. So in terms of application, These are just two examples that I found interesting. Ion exchange chromatography is used in water purification, basically the softening of drinking water. It's also used in the separation and purification of blood components in the medical field. So in terms of advantages and disadvantages, I'm going to start off with the disadvantages because I kind of want to end on a high note. So a buffer is required for ion exchange chromatography to work. This is also a process that only uses charged molecules. So if it doesn't have a charge on it, it essentially can't be used. To move on for the advantages, this is one of the most efficient methods of separating charged particles. It can also be used for many different charged molecules, like as I said before in the beginning, large proteins, nucleotides, amino acids are all fair game for this. It also gives us a high yield as well as concentrating the sample. So ion exchange chromatography is just really useful. It's one of the most efficient ways of separating charged particles. And these are my references. And thank you guys for listening.